I thought I'll share my experience with the group on what are the challenges in building capacity and culture in intensive care research based on some basic science work and one large clinical trial that I ran, and currently doing 4 COVID-19 trials in India. And I think each of these has taught me valuable lessons, which I'm very happy to share with you. So, so this is going to be more of a narrative review. There is sort of no hard science to talk about at this stage. I mean, it's a lot of science, but no, I'm not going to go through any data in detail. And I'll share my lessons from my personal experience and mainly from the 2 trials, the adrenal trial and the COVID-19 work, and what lessons can the younger people in this audience they can take from this. These are my disclosures. And so I trained in India. I trained in a large tertiary center in Vellore. And then my background is physician training, anesthesia in the UK, where I worked for 2 years developing a continuous blood gas monitoring system. And then I migrated to Australia in 1995, and I did my ICU training here. So this was the device that we worked on. So this device can measure pH, PCO2, and PO2 continuously. So when this device was developed, it worked very accurately. You could place it into an arterial line in critically ill patients. You didn't have to take samples of blood out from the arterial line for blood gas analysis. This gave a continuous reading. It reached, it got FDA approval, reached clinical application, but the problem was with the company, when they marketed it, made it too expensive for hospitals to buy it and use it. So the cost of a simple blood gas analysis costs about $5 in Australia, but to buy to a sensor would cost 1,000 Australian dollars, and therefore it really wasn't economically viable. So the device went out of the market. The Royal Brisbane Hospital in 1995 had bought 2 of these devices and did not know how to work them. So they advertised for a fellow who would take on this role, and I applied for the job and I got it. And so I came here and I thought, okay, I'll set up a program of work with this. Now, it wasn't easy, despite several publications and despite me being the most experienced person with this device, it wasn't easy. So I had to establish my credibility with the group here. And in 1995, there was minimal ICU research. John Morgan was my mentor, and he had done a lot of work on acid base. So a number of things which this device could measure, there was a convergence of interest because he was interested in acid base. So I was given the sluice room in the neurosurgical ward to set up the laboratory. So we set up a small laboratory that's called a tonometer, where you can measure gas tensions, and we set up a tissue oxygen lab there. And in the next couple of years, we flooded the Royal Brisbane Hospital Symposium with our abstracts. We won several awards, and then people started to notice our work. And then we went to the ANZICS meeting, which is our Intensive Care Society National Forum, and we got prominence in national meetings. And so Queensland and certainly Brisbane, for intensive care anyway, began to get noticed. Now, then we had to build a profile and make sure that more people got to know us. So at that time, my interest was still basic science. So we set up a critical care basic science symposium, which ran for 3 years, and we had a number of fantastic basic science researchers internationally who came and spoke to us. But then again, at that time, lots of meetings were happening in intensive care, and there wasn't room for this meeting to continue. Lots of clinical meetings had a component of basic science, so people realized they weren't getting their bang for their buck coming to a basic science meeting. So we had to stop running this symposium. So and then around the same time, in clinical intensive care, corticosteroids became very popular for people with life-threatening sepsis. And the premise at the time was that these patients had what's called relative adrenal insufficiency. The adrenal gland did not produce sufficient cortisol for the amount of stress that they were exposed to. So, and I personally, had a lot of interest in adrenal cortical work, so I did not believe this concept. So I did a lot of basic science work to disprove this hypothesis. which provided sufficient foundations for me to run a large clinical trial. And basically, and that changed the way we could run the clinical trial design, because the steroids and sepsis was a question which had been dogging clinicians for nearly 50 or 60 years. So we commenced basic adrenal endocrine research. We got all the data, and then we surveyed the clinicians, and they were very happy to take part in a clinical trial. Now, this was the trial. This is the adrenal trial that we led, which was published in the New England Journal of Medicine. It was the world's largest septic shock trial, and I was privileged to lead the trial and be the first author of this paper. And this trial took 10 years from conception to completion. Now, the stories, and this trial was obviously recognized worldwide and by the New England Journal editors as one of the top articles of 2018. It was the final trial, the Australian Clinical Trials Alliance, the ACTA. We became one of the finalists for the award for that, for ACTA awards, and we also won the statistics award in the ACTA awards group. Now, this story sounds fantastic. It all sounds like it's all straightforward. We had no troubles and so on. Let me share the troubles with you. Let me share the pain with you. Now, I conceived the trial in 2005. And so I applied to the NHMRC in 2005, unsuccessful. I applied in 2006, unsuccessful. I applied in 2007, unsuccessful. I applied in 2008, unsuccessful. I applied in 2009, unsuccessful. So the most important lesson there is, if you believe in it and you've got the science right, then you should not let the proverbial grind you down. So you just keep persisting. And so in 2010, we finally cracked it. So we got the NHMRC grant. And so the trial really took, as I said, nearly 10 years to complete. And it was run in 5 countries, 69 hospitals in 5 countries, and we enrolled 3,800 patients. Now, just for you to be aware, so intensive care is a very difficult environment to do research because patients can't provide their own consent. They're often unconscious. The families are under a lot of stress. And so we worked through all those issues, as well as and there were a number of regulatory approval issues, which Michael referred to in the previous talk, and I'm sure it'll come up during question time, and I'm happy to talk about that with you as well. But so we had to work through all that, and there was a lot of personal time that I had to commit. My wife called herself an adrenal widow for the duration of the trial. I had to use my long service leave, applied for leave without pay, and bore a number of travel costs as part of this. And you're on call 24/7 for the entire duration of the trial. This is what happens. People will phone you from any part of the world if they have a question about enrollment, whether to continue the trial drug or not to continue the trial drug, they would phone you. We had a trial manager, but we still had to be available. And you have to keep sites engaged and avoid trial fatigue. And that's a huge challenge when you're running a trial over a 4 to 5 year period. And so you send them regular emails and congratulate them on every randomization that they do, newsletters and SMS messages. So, so every day I would send a message to all the colleagues in Australia. I know it's your weekend Monday on call, but do not forget adrenal. Please keep enrolling patients. But that's what you need to do. You have to really immerse yourself to get the trial completed. So I mean, it got to a stage where my colleagues at the PA hospital set up a special ringtone to recognize my calls. And so they knew Bala was calling. And so they were very, very careful. Anyway, so, and that's about adrenal. And just let me share what happened with COVID-19. Now, obviously the last year has been consumed by COVID-19, and it's changed many people's lives, personal lives, professional lives, research. And I just want to talk to you about the burden of disease in low middle income countries where I know there's been a lot of talk about the caseloads in the US and Europe and how things are. And I just want to give you a bit of perspective of what happens in LMICs. So this is the top 10 countries in the world. These were the numbers as of yesterday morning. And USA still has got about 30 million infections, and Brazil is about 11.7 million. India is 11.4. Now, these are based on RT-PCR testing. This is a study which was published in the JAMA, a newsletter, a research letter, which looked at prevalence in one particular state in India based on antibody testing. And there have been several such studies amongst hospital staff, amongst the general public, in villages, in towns, in cities from different parts of India. And the message is very, very clear and consistent. Somewhere between 25 and 40% of the cohort are testing positive for antibodies. This is pre-vaccination. So what it means is if you extrapolate that to the entire Indian population, that means somewhere between 250 to 500 million people have been infected with COVID in India alone. So if you're doing a COVID-19 trial, and particularly you want to have, if you want to have an international trial, generalizability, and looking at relevance across different healthcare systems, then one of the LMIC regions you really look at is in India. So in April 2020, we got funding from, we applied for funding in Australia to do a hydroxychloroquine prophylaxis trial. And we were picked to the post by the University of Melbourne. And so they had a trial funded, we didn't. So we got funding from the Wesley Medical Research, a small, a pilot study to do a 6,000 patients, 6,000 healthcare worker study. And this was what's called the HOPE trial, which we ran through the George Institute for Global Health. There's an office in New Delhi. Then in May 2020, the ASCOT trial, which is the Australasian COVID-19 trial, which is funded by philanthropy, and this was meant to be a factorial design study. For those of you who are not familiar with a factorial design study, is where you test intervention 1 in one arm, intervention 2 in the other arm, then intervention 1+2 in the third arm, and standard of care in the fourth arm. So that's in a standard RCT, you compare intervention 1 versus 2, or 1 versus control. In a factorial design, you can have multiple interventions. So and it started as a factorial design study, but then it had to be moved on to an adaptive design. And again, COVID-19, thankfully, the numbers came down here. And then they approached us to see if we could do the trial in India. So we took on the ASCOT trial. And then again, our colleagues in Denmark, who are our collaborators in the adrenal trial, they came to us and said, look, we are trying to do a steroid study in COVID-19, and could you please help us out with finding sites in India? And so we took on the COVID-19 trial, the steroid trial there as well in May. And then a Belgian company had developed a mask, which a 3D printed mask to minimize the need for mechanical ventilation. And mechanical ventilatory support was at a premium, and the devices, there weren't many devices in resource limited settings. And therefore they said, could the George Institute in New Delhi take this on and see if you could use it, and if we can minimize the need for mechanical support in COVID-19 patients. So we took on the trial. So with 4 trials, we had a task cut out. There was a lot of hope and expectation. There was a lot of excitement there that we were taking on 4 trials, and there were 30 to 40,000 new cases per day at the time in India. And so we thought we'd be able to complete the trial in about 2 or 3 months, given the large numbers of cases. Of course, we were wrong. So how did we go? So we knew there were going to be resource issues in India, because clinicians over there do not get non-clinical time as clinicians in Australia. They are 100% clinical all the time. And so that's a challenge. And the research infrastructure is not well set up as it is. We have the luxury of a fantastic research structure here. They don't have it there. And so the first thing was, we also have multi-site ethics approvals here. They don't have that. They don't have the system there. However, during COVID, the Indian Council of Medical Research said you can allow multi-site ethics approvals. But for that to percolate through to the individual hospital ethics committees took a long time. And so it never actually happened. So we had to get individual hospital ethics approvals from different for various sites. And then for overseas studies, you have an additional regulatory approval, which adds another 2 months. It has to be screened by the health ministry, which had to do that. India has got 28 states, and therefore there are 28 languages. So the patient information consent form had to be done in 28 different languages. And because clinicians are over here, when you do a clinical trial, you say, look, there's going to be a startup meeting on this particular day at this particular time, Zoom in, or let's all meet up, but you couldn't do it there because clinicians are all busy. So you ended up having to do a startup meeting for each site, for each trial. But that's the way, that's how we've got to work, you've got to do it. Despite all this, we commenced the trials. The HOPE trial commenced in June 2020, the COVID steroid in October, the mask trial in November, and the ASCOT trial. It took a long time because there were multiple changes of protocols, and it only started in February 2021. As soon as, so with the hydroxychloroquine trial, I mean, let me explain this to you. So the hydroxychloroquine is a prophylaxis. So we want to see if hydroxychloroquine, given as a prophylactic agent, will prevent COVID-19 in healthcare workers. And therefore, the testing was the PCR testing. So, but the Indian Council of Medical Research issued an advisory based on laboratory evidence saying that, in fact, we recommend that healthcare workers take hydroxychloroquine. And so when that advisory came, we could not enroll anyone into our trial. It became a challenge. So this then became public news, and there was an editorial in the BMJ, where the medical fraternity complained about the ICMR's recommendation, and even the George Institute director, he said, look, we are actually doing a clinical trial. Could you please use that as the evidence base to run a trial? And the ICMR wouldn't, it all fell on deaf ears, so we couldn't do that. But we used that opportunity to, obviously, the whole team which was assembled to do this, we were able to work out the surge capacity of Indian critical care units during the COVID-19 pandemic. What we found at the time was that the PPE, based on the available data, there were only 334 PPE sets across in hospitals in India. I mean, that's nothing, but that's all they had. And therefore, there were implications for consent, physicians wearing PPE and meeting healthcare workers to get consent. And so it became a huge, and also we were also comparing hydroxychloroquine plus PPE versus just standard PPE. And therefore, getting standard PPE across all hospitals in the same way was not possible. There was a lot of stigmatization if you tested positive with COVID in India at the time. And so healthcare workers were reluctant to get tested if they had symptoms, because once you got stigmatized with that, then your landlords would say your tenancy is cancelled, please leave our homes. And so there were many cases of those things happening there. And finally, in January, when the vaccine came in, India had its own vaccine called the Covaxin, and one of the advisories was that use of chloroquine concomitantly will impair antibody response. So in people who had been randomized in December and January, where they had to complete the 12-week course, when they were going to get their vaccine, then they had to stop taking chloroquine. So a number of factors, obviously, we had to navigate. But despite that, we were funded by the Wesley Medical Research. And compared to the other large groups which had received funding, our funding was nowhere near that. They were in millions of dollars. But the Kopkov trial from Moru, the Mahidol University in Thailand, they only enrolled 226 patients. The Walter Eliza group in Melbourne, which was funded $3 million, they enrolled 270 healthcare workers, and we still managed 419. Significantly less than what we had expected, but that's what it is. The COVID steroid 2 trial, we are nearly done. We've more than 70% of the way there. We've enrolled 715 patients. The mask trial, we are 25% of the way. And the ASCOT trial, which only started last month, we've enrolled 90 patients. So why did we succeed? So the question really is, why did we succeed with COVID-19 trials? I think we have to respect and accept differences in system. We have to accept the way people work. That's the way things work in a different environment. We have to work with them. We have to work with them as equal partners. It is not our trial and they're doing the work for us. It's in fact, they're all part of the trial. And so you have to allow ownership of the trial by them. And we were very keen, in all these trials, we made sure, certainly in 2 of the 3 trials, the first and second authorship of these papers were going to be our key Indian collaborators. And there was, and obviously we mentored them, and one of them in fact won the Young Investigator Award in their hospital system, which is very gratifying. And importantly, also to mention, with the adrenal that I spoke to you about before, there were 11 secondary publications which came from adrenal, and 8 of them have early career researchers as first authors. And I think that's important. It just needs to happen. Others feel that they are being recognized and they're able to grow. So when you do work, research, it's not easy, but patience is important and you've got to persist. Even if they knock you back for grants 5 years in a row, apply in the 6th year, don't stop. Engage with people, you deal with frustrations, and you've got to work with people because people are working for you. And remember, when you run a large trial, after 4 years of work and lots of sites, lots of people enrolling, it is your name which is going to be the first author. And therefore, you have to recognize that it will be known as your trial. And therefore, you have to appreciate that, and so not get frustrated with people if they don't enroll patients. And you've got to have good colleagues, have a good team, and lead by example. So you've really got to be the face of the trial for the entire duration. And I want to talk about persistence. So this is the Framingham trial, and this is important because the Framingham trial Framingham is a small region in Boston where they worked out the epidemiology of cardiovascular disease. And they started their work in 1948, where it was the whole idea was conceived. In 1956, they worked out the risks for rheumatic heart disease. In 1959, for ischemic heart disease. Then they worked out in the 60s the relationship between smoking and heart disease, and then exercise, lipids, diabetes, AF, and stroke. Systematically, they worked out step by step. And now, in 2002, the grandchildren of the original cohort are now being followed up. Now, this is a program of research. 50 years, it doesn't happen overnight, 50 years, but obviously someone started it and people are continuing that, but it continues to inform us every day. And collaboration is important. And with collaboration, because you obviously build up your own CV as part of that, you start to write the appropriate sections of the grant where you work with people, you build up a track record, and it improves the science and the feasibility of your own project. And it's important to be open to ideas, avoid tunnel vision. And this is a very good sociology paper, which is it's called the mundanity of excellence. What it actually takes to achieve excellence. And they looked at Olympic swimmers, and what they did was every morning they got up and they just got up and jumped in the pool and swam for hours together. That's all they did. And the secret to excellence is the degree to which you successfully maintain mundanity. You just come in and you do your lab work every day. Keep your records in detail, follow the protocol, follow the methods, and you will succeed. So in the end, to conclude, there are plenty of opportunities for research, and the potential for research is now much better than it was 25 years ago. And you will face challenges and obstacles. It is not going to be easy, but they are not insurmountable. Talk to people and they will help. There is, at the end of it, you'll have immense professional satisfaction. It's a rewarding experience, and you'll have the satisfaction of solving a scientific question. But most importantly, you will improve patient outcomes. Thank you.